At the turn of the century in Britain, the average life expectancy was 45. Infant mortality was 20 times higher than it is today. A constant fear in people's lives was of the germ and the microbe. No one understood much about them, but they brought death. Hospital wards were full of those suffering from septicemia, tuberculosis and pneumonia. Epidemics were frequent. In 1918, a mutant strain of the influenza virus swept across the world with devastating effects, killing 25 million people. It was known that some disease was caused by bacteria, single-celled creatures which could only be seen under the microscope. One could become a million in 12 hours, and their toxicity caused tuberculosis, cholera, plague. But for yellow fever, polio, influenza, there seemed to be no agent of disease at all. No microbe could be detected under the microscope. When scientists looked down the lens, all they could see was devastation. These are human cells under attack by an invisible enemy. It kills cells with stunning efficiency. For lack of any other information, it was named after the Latin word for poison, virus. What was in this mysterious liquid that could kill? A deadly chemical? Some strange germ? No one knew until in 1932 the Germans opened up a whole new world for science with the invention of the electron microscope. It used electrons to show up objects, although the objects themselves were killed in the process. With it they could peer into a universe smaller than the wavelength of light. It was in this universe that the viruses lay. These are hundreds of viruses gathered inside a living cell. It's the only time viruses have ever been filmed. This unique footage, shot in 1966, took a year to make, in a vibration-proof room using time-lapse photography and focusing on one of the biggest viruses, vaccinia, which hovers on the edge of the visible. Viruses have no life outside a host. They lie dormant until they're sucked into a living cell, the cell of a plant or animal, and then they too apparently magically come to life.
I believe that viruses probably go back to the origin of life. They're chemicals that can replicate. They're the simplest forms of life. The reason viruses cause disease is not that they set out to do that. They set out to survive. In the process, they must grow in cells. When they grow in cells, they often kill the cell, and that damages cells within your tissues and organs, and you get what we call disease. Or in some cases, in rare cases, they cause cancer.
A virus is a simple but strange organism. Just a strand of genetic material, DNA or RNA, wrapped in a protein coat. On its own, it can neither survive nor replicate. It's just a tiny clump of chemicals. When it enters its specific host cell, it takes over, grabbing spare chemicals from the cell itself and linking them together to make clones of its own unique genetic pattern. Hundreds of these clones then bud out of the cell, ready to infect others.
Because they are part of us, we can't intervene with antibiotics as we can with bacteria. Bacteria are separate living cells. The virus is not a separate living cell. It becomes part of your cells and my cells. Now, in order to intervene, to make an antiviral chemical, antiviral agent, we must go in there and sort out all that DNA and RNA inside the cell and say, I'm going to zap this little piece right here and nothing else. I'm not going to damage your DNA at all. A very tall order, very difficult.
Our only remaining serious competition for dominance of the planet at this time really are the viruses. And it has to be said that our own survival is not necessarily preordained.
The question of the origin of emerging viruses might have remained the obsession of a few scientists, but for a tragedy that slowly began to unfold 10 years ago. It has focused the whole world's attention on emerging viruses and galvanized the scientific community like nothing else before. The disease was first recognized 10 years ago in America, but by then it had already struck Africa, where the epidemic is now of frightening proportions. 6 million are probably infected. AIDS, a devastating breakdown in the body's immune system, can be caused by 2 viruses, either HIV-2 or the more widespread HIV-1. The viruses attack T cells, the very cells that alert the body to defend itself against disease. Nothing like this has ever been seen before.
The quest for a cure is really a search for a weak point in the life cycle of the virus. The AIDS virus attacks these cells, the T cells that live in our blood and coordinate our defense against disease. A protein on the surface of the virus locks on to a special receptor on the surface of the T cell. The virus then injects its genetic core, leaving behind fragments of its coat on the T cell's surface. Some of these coats then seek out other T cells. They act like glue, binding the T cells together in large clumps. After a few days, the diseased clumps burst apart and die. The effects of this initial attack are soon visible. These are healthy cells. This is what the AIDS virus does to them within just 48 hours of infection.
In contrast to what people in general believe, immediately after that the virus enters the body, the virus immediately starts to attack the immune system. And it doesn't sit there for 10 years and waits for its chance, but it immediately starts a sort of war against the immune system.
T cells are vital to the body's immune system. Scientists have just discovered that when the AIDS virus enters the body, the T cell count falls to virtually 0. The immune system is nearly destroyed. But the body fights back, and for reasons not yet understood, the T cell count returns almost to normal. But the surviving T cells are not normal. The genetic core of the AIDS virus remains buried inside them. The infected person may appear healthy for as long as 10 years. But sooner or later, the virus overwhelms the body as it reproduces in ever larger numbers, hijacking the machinery of the cell, and in effect, turning it into a huge AIDS virus factory. The AIDS viruses cover the dying T cell and escape into the bloodstream in search of new T cells to infect. As at the moment of initial infection, the T cell count again begins to drop and the immune system is wiped out. Now the person has full-blown AIDS. The only approved treatment for the AIDS virus is the drug AZT, but AZT has severe side effects. It kills blood cells, requiring some of those who take the drug to have transfusions. But even if AIDS patients could cope with the side effects of AZT and DDI, these drugs are flawed in another more fundamental way. They fail because the AIDS virus is constantly changing its nature or mutating.
Viruses, in general, change as they grow. We don't think of a virus anymore as one individual item anymore as we think of human beings as individuals. We know that it's a wide range of what we call human beings. We all look differently from one another. We speak differently. And the same thing with the virus. There is something called an AIDS virus, but no single AIDS virus is identical to any other AIDS virus. And the faster it replicates, the more variation occurs.
As the AIDS tragedy has deepened, scientists have intensified their efforts to find a cure. Some of them decided that an entirely new strategy was needed, one which would focus on the coat of the virus and on the mechanism by which it attacks and penetrates the T cells.
The coat of the virus is crucial to its function. It has to bind to and enter cells, and the outside of the virus is what determines that. A protein called the envelope protein is required for docking to cells and for entering cells.
So the scientists began to study this docking mechanism in more detail. They discovered that there was something else at work on the coat of the AIDS virus.
We found several surprises when we looked at the structure of the AIDS virus. Rather than being a simple protein, we found it to be very heavily modified by complex carbohydrates, these sugar molecules. Over half the size and weight of that protein turns out to be sugars.
In Britain, scientists at Oxford University were well aware of the potential significance of this finding. For Oxford is a leading centre in a new science, glycobiology, the science of sugars. This new science is helping us to understand the role of sugars on the surface of cells, and in particular on the way sugars help viruses attack their targets.
People have always known that sugars were attached to protein molecules. But before 1985, we didn't really have the technology available with which to study these sugars.
During the past 5 years, Raymond Dweck's department has developed complex machines which isolate sugars and analyse them to build up a picture of what they look like. This is a computer simulation in slow motion of what goes on at the surface of cells. The static part of the picture is a bedrock of protein. On its surface are sugars, which rotate billions of times a second. It became clear that these sugars are far more important in the body than scientists had ever realized. The sugars project out from the protein on the surface of the virus and are involved in locking on to the T cell. Could it be that a particular configuration of these sugars is what gives the virus its ability to enter the T cell? In this lay the germ of an idea that was to present an entirely new strategy in AIDS research.
The sugars on the surface of the virus do help the virus find its target. If we could interfere with those sugars in some way, then we might be able to stop that virus finding its target. Now, one way of trying to interfere with those sugars is to interfere with the very process that puts them on, the biosynthesis of those sugars. And if we could find molecules that might alter that in some way, we would produce abnormal viruses which would then not be able to find their targets.
The answer was to come from a surprising source, Kew Gardens in London, home to a wide range of plants from all over the world. Within its walls also are scientists dedicated to finding out what chemicals these plants contain. Linda Fellows is one of those scientists. She has for years studied compounds from plants that have the ability to deter predators. Back in 1977, she discovered some new compounds. They came from a family of such plants found in countries as far apart as Australia, Japan and Brazil. When she analysed their chemical structure, she was puzzled, for they were not what she had expected to find.
It was only when we looked at these compounds in a completely different way, we just simply turned the molecule round, that then we realised in a flash of inspiration that they are very, very similar to sugars.
Locusts are notorious scavengers, and given the opportunity to eat sugar, they do. Linda Fellows wondered whether they would have the same appetite for her family of new compounds, which she called sugar mimics. The sugar mimic was placed on the locust's taste buds. Analysis of the reaction led to the conclusion that sugar mimics disrupt the processing of sugars. As Linda Fellows' interest in the new compounds developed, she decided to find out more about them.
I began to look around through the scientific literature to see if anything similar had been discovered, and I was intrigued to find that almost at the same time, this was back in 1977, a Japanese group had reported a very similar substance in mulberry roots, and they'd been chasing an agent with anti-diabetic activity.
The Japanese use a sugar mimic as an anti-diabetic remedy in traditional medicine. This compound is believed to interfere with sugar absorption in the stomach. Excited by her discovery, Linda Fellows decided to test the compound for herself. In his laboratory, Stan Timms began to test the effects of the Kew Gardens sugar mimics on the AIDS virus.
After I met with Linda, she sent me some compounds for us to investigate. We were very excited about this thought because at the time these molecules hadn't been that well investigated, and they were new because they were natural products.
Human cells are grown in small tubs in the laboratory. Under the microscope, healthy cells look small, round and even. But when the AIDS virus is added, the telltale signs of infection quickly show up, and the diseased clumps appear. What would happen if a sugar mimic was added? Stan Timms was amazed when he found out. In culture after culture, there was little sign of the diseased cell clumps. The cells looked healthy. It seemed some of the Kew Gardens compounds could kill the AIDS virus in the test tube. Had Stan Timms really stumbled across the magical drug the world had been waiting for? A cure for AIDS?
When Max Perutz invited Linda Fellows and Stan Timms to come to the MRC committee, and they presented their findings, we were actually very excited.
Excited though he may have been, he was also highly sceptical. Back in his laboratory in Oxford, he was worried that if the compound was harming sugars on the AIDS virus, it was almost certainly deforming them on everything else in the body as well.
You see, all our cells are covered in sugars. And what we want to do is only affect those sugars on the virus. We don't want to affect the sugars on all our other cells as well. That's why it's important to find something that's really selective only for the virus.
So the next step was to gather together a collection of sugar mimics and to send them to one of Britain's most specialised virus testing laboratories. The sugar mimics were sent to the first scientist in Britain to have isolated the AIDS virus, Abraham Karpas. At his laboratory in Cambridge, he took on the job of finding out if any one of the sugar mimics would not merely kill the virus, but do so without damaging human cells. He knew how important it was that any anti-AIDS drug prove not to be toxic.
When Abraham Karpas sent back the results of the first 17 or 20 compounds he tested, they all looked pretty bad. The idea seemed to have failed.
The compounds had failed because they were toxic. As Raymond Dweck had feared, the sugar mimics seemed to be disrupting the body's own complex sugars indiscriminately.
You're really up against it, because everything is covered in sugars. That's the problem. And I think a lot of people felt that we weren't going to go any further.
I was beginning to wonder myself about the future of these compounds as drugs. If we were going to have to use such an enormous concentration, I felt there were bound to be toxic side effects, and it would be too expensive, and the future didn't really look that promising. So I began to wonder if there might be ways of modifying these compounds.
Linda Fellows had the idea, which turned out to be a very brilliant idea, to use different derivatives of the same basic compound for the reason of penetration into the cells.
The idea was to modify the drugs chemically so they could penetrate the cells more easily. Then low doses of the sugar mimics would be less toxic, but still be effective against the virus. Drawing on the expertise of the university's chemistry department, the Oxford unit manufactured dozens of new compounds. Each was provided with a distinctive tail designed to help get it into the cells. A further 19 different compounds were synthesized in the laboratory and dispatched to Cambridge for testing. Compound number 57, butyl DNJ, was strikingly different to the ones that had gone before. It completely stopped the AIDS virus from infecting new cells, and it seemed to have no noticeable toxic side effects. Abraham Karpas found that butyl DNJ gradually eliminated the infected cells from the culture, He removed the compound and watched the cells for a further 100 days. No virus reappeared, and there was no sign of toxic effects. The compound extracted from the mulberry tree at Kew Gardens and modified in Oxford had triumphed over the AIDS virus in the laboratory. Butyl DNJ had emerged as a leading candidate in the quest for a cure for AIDS. It blinded the virus, it wasn't toxic, and the beauty of it was that since it could deform any AIDS virus, it would overcome the problem of mutation. Searle took steps to mass-produce butyl DNJ in the company's Chicago factory and to put the drug through the many tests necessary before it could be given to AIDS patients. The new compound was first given to laboratory animals, mice, rats, dogs and monkeys. The principal side effect was diarrhea, which Searle believed would not limit the use of the drug. Butyl DNJ was soon shown to be safe enough to be given to humans.
Ordinarily, in other drugs, that kind of a process can take anywhere from 3 or 4 years. In the case of butyl DNJ, we moved from the observation in the laboratory to the first clinical test in less than 1 year.
Amid great secrecy, Searle won official approval to give the compound to AIDS patients. The company then tried to find individuals who had not been on AZT so that it would be possible to isolate the effects of the drug. Among the first patients the company recruited was a scenic designer from Boston, Massachusetts, George Jones. He got AIDS 2 years ago, yet he was prepared to forgo the benefits of AZT and its proven ability to prolong the AIDS patient's life for the uncertain effects of the new drug. George knew that the dose he would get would be too low to cure him, and there was a chance of adverse side effects. Yet he agreed to take it.
When I had pneumocystis, two proven drugs didn't work, and I had to undergo an experimental therapy. If someone before me had not gone through a protocol with these drugs, I wouldn't have the benefit of taking them. So there were people who had gone before me who were brave enough, who were willing enough to give their time and energy to go through these protocols, to develop these drugs, and I benefited from it, and thank God someone did.
But unfortunately for everyone living with the spectre of AIDS, there are all too few people willing to take the risks George Jones took. Last March, it was reported that Searle's clinical trial was frustrated because the company could not find a sufficient number of people to take the low doses the test required and to keep off other drugs. The company is finding suitable patients, though slowly, but it is naturally cautious about revealing results prematurely or making any public claims for the drug.
People in the research and development business are always hopeful, but you have to realize that most compounds are not successful, even at this stage. The industry average of the number of successes of compounds that are at this stage of understanding is probably no greater than 25%. We're hopeful.
There are no guarantees of success in science, and it's quite possible that the new strategy may fail. Despite all the effort and hope of everyone involved in the discovery and development of butyl DNJ, the AIDS virus may yet prove to be an even more formidable opponent than anyone had thought. Until the quest is over and a cure is found, the AIDS epidemic will continue to claim more and more lives.